All right, I think we'll go ahead and get started. So good evening, everyone, and welcome. Thank you for joining us for the program New Approaches for Treating Childhood Leukemia and Lymphoma. My name is Lily Evangelista. I am the Patient and Community Outreach Manager for the Leukemia and Lymphoma Society in the Greater Los Angeles region. So this program is part of the Closer to a Cure community lecture series brought to you in partnership with UCLA Health and the Leukemia Lymphoma Society. So we are very excited to have you tune in today. So right now, we're just gonna do some quick housekeeping before we get started on the program. First, I just wanted to highlight and make sure that you knew where the chat features were. If you have a question for our speakers, just go ahead and click on there and type and send your questions, and I will be reading them out loud during the Q&A. In the event that you also have any technical questions, please go ahead and communicate through there as well. We are recording the program today, so for your reference and to help others in the future. So to ensure we respect individual privacy, we will only be recording the presenters and their slides, and we'll have participants muted during the recording period of the program. Okay. All right, so very quickly, I did want to go over a little bit about the Leukemia Lymphoma Society. So we are a nonprofit organization focused on finding cures for blood cancer, but also providing support for patients, survivors, and family members navigating a blood cancer diagnosis. We do have an information resource center that is filled with specialists who are there to help you to navigate any of the resources that we have at the Leukemia Lymphoma Society, but also to answer any of your disease or treatment questions. And they can easily be reached at this phone number, at the 800 number, or you can also email individuals as well as go on our website. So when you reach out to one of our information specialists, we, like I said, do personalized disease treatment and navigational support. It includes helping go through clinical trials and nutrition. We also have a lot of educational materials. So for example, we have a ton of booklets on specific diseases, but also things like we have a caregiver workbook, we have a living and learning and how to advocate for your child's education. We also have a new coloring app for kids, which is available that you can utilize. We do have peer support as well. So if you wanted to contact or be connected with other individuals who might be going through the same thing, other parents, we can connect you one-on-one, as well as we have online support groups going on at the moment. For financial assistance, we do have the Urgent Need Program, which is specifically for our pediatric patients. So please get in contact with us if you're looking at any of these resources. Lastly, we know that it's really hard to stay connected right now, but we have a ton of other things that you can get involved with in the organization. We have numerous campaigns like coming up on November 7th. For those who are in Los Angeles, we have our Light the Night campaign virtual experience. So normally we would gather and have a walk, but this year we will be bringing the cancer community together virtually to celebrate and commemorate all those who've been affected by cancer. So it's a wonderful opportunity to kind of come together online. And we're really excited to have UCLA Health to be a presenting sponsor for this year's experience. So if you would like to register, go ahead. We have lightthenight.org and you can sign up through there. We also have a ton of volunteering opportunities. We're always looking for people who are inspired to share their story, to support other families, but just to inspire people towards the mission. So without further ado, I'd like to introduce our speaker. He has been a pediatric hematologist oncologist for the last 2 decades, and he's currently at UCLA Mattel Children's Hospital. So Dr. Sotero de Oliveira, please go ahead and take the floor.
Okay, can you see my slide? Yeah. So, hi, good evening. I'm Satiro de Oliveira, and today we are going to discuss about a few updates on childhood blood cancers, in particular acute lymphoblastic leukemia, that is ALL, acute myeloid leukemia, that is AML, and Hodgkin's disease. A little bit about me. I'm associate professor in pediatrics at the Division of Pediatric Hematology Oncology. And I'm a part of the stem cell transplant program and the cellular therapies program. My clinical focus is on the transplant, bone marrow transplant, stem cell transplant, and gene therapies. And I do research on integrating then cellular therapy and gene therapy to harness cancer immunotherapies, in particular for hematological malignancies. I'm kind of middle-aged, so I graduated in 1995. And over these 25 years, I've seen a lot in both countries, Brazil and the US. And I'm going to share a little bit of the advances I've noticed over the last 5 years at least. So what is the status of childhood cancer in 2020? This is directly from the American Cancer Society. That is about 11,000 kids in the US will be diagnosed with cancer in 2020. And about 1,200 children are expected to die from cancer in 2020. From the website, from the St. Patrick's Foundation, they have that up to age 20, about 1 kid in 285 children in the US will have cancer. And worldwide, 1 child is diagnosed with cancer every 2 minutes. Also from the American Cancer Society, we have that about 84% of children with cancer will be able to survive their disease 5 years or more. This means that this is different from the cancers we see in adults. There's always hope, and the chances of cure, they are way higher, mainly because the cancers that arise in kids, they are more immature and they are more sensitive to chemotherapy. And because of that, the focus is not only killing the cancer at any matter, but also leading the kids to a very productive and healthy survival. So there's no better example of all these advances, and this is actually one of the most successful stories in the whole field of medicine, is the history of the treatment of pediatric ALL, that even though it is a rare disease, through collaboration between multiple institutions, and in particular here in the United States, congregated by the Children's Oncology Group, or COG. So based on a series of different clinical trials, they were able to learn what worked best and build upon that, launching successive clinical trials to improve the treatment. And nowadays we have improved a lot, to the point that a child coming through the doors with a diagnosis of ALL, we can expect that about 95% of these children will be able to be rescued with only chemotherapy, without any other problems on their therapy. And this is here, you can see it's years from diagnosis and survival. Most of the hardship is in the beginning, because essentially that is when they are still actively fighting the disease and receiving the toxic chemotherapies. And after that, the disease may come back. That's why these lines are not completely steady. So this is the concern, the idea that we can cure them and promote health and not allow the cancer to come back. So now focusing a little bit better on ALL and also non-Hodgkin lymphomas. As we were discussing, these diseases are rare if you compare to, for example, breast cancer and prostate cancer. So about 3,000 new cases a year in the US for ALL, about 800 new cases a year for non-Hodgkin lymphoma, considered as a whole group. And we are going to get about 85 to 90% of them being cured, but we still have a huge number of kids who will perish, because the problem is, for those who cannot get a proper response, or if the disease comes back, we will have less than 50% of chances of cure. This is why it's important for every child with suspected diagnosis of cancer to be referred to a center specialized in the treatment of childhood cancers and integrated at the COG, so that way they can receive the best of care. A lot of different pediatricians may never be able to diagnose childhood cancers, either because they simply they have never seen it because it's so rare, or because they missed the diagnosis. One very important concept we learned from all these different clinical trials is the concept of minimal residual disease, or MRD. And this, what we have learned from all the kids with cancer, has now, after so many years, been translated even for the treatment of leukemias in adults. The idea is that if you're able to eliminate every single leukemia cell of the body, or at least limit, decrease or kill the great majority of them, to the point that you cannot detect any minimal residual disease in the bone marrow or in the peripheral blood, can really be significant for the treatment of these kids. Like here again, you can see time along years, and this is the survival probability. So if you can, after the first month of chemotherapy, be able to not to detect any MRD on the samples from these patients, they have an outcome that is completely different from those patients who, for whom we can still detect some residual leukemia. To the point that then, after the first month of chemotherapy for ALL, you can already have a pretty much good idea if you need to be more aggressive, or if you even need to switch treatment because it's not working properly, or for those whom you can decrease the toxicity and not only aim at survival, but also to be less toxic and promote faster recovery. So thinking of talking about MRD determination in acute lymphoblastic leukemia, what has been now the standard over the last decade is the use of the determination of MRD using flow cytometry. It is a machine that will count all the different cells by defining them, what they are, if they are leukemia cells or if they are normal cells. And this machine, the flow cytometry machine, is able to detect one leukemia cell in 10,000 cells to 100,000 cells. And throughout all these different clinical trials, we have learned the prognostic value. So in a way that if you have the determination, you can even quantify and define the chances of cure for a particular patient. And over the last 3 years, what has come up with the developments of new technologies and studying way more patients is that there's a different method for the determination of the MRD, that it is through the sequencing of the DNA. So that way, if you target different genes, in this case, either the immunoglobulin A gene or the T-cell receptor gene, then you can have a higher sensitivity to detect the leukemia cells. So then you can detect one leukemia cell in about 1 million of cells that come. This is not possible in all patients. It's only about 96% of the cases. And this is harder to standardize through different institutions. That's why this is still a matter of further studies. That's why it's not as established as the MRD by flow cytometry. And here I'm bringing some very interesting data to show the power of these new technologies and how the MRD determination really matters for the therapy of these patients. So here, what we're talking about is along years, the event-free survival is in essence, the survival without the leukemia coming back. So here you can see in black is the MRD determined as by the high throughput sequencing, so the HTS, the DNA sequencing, and in green, the determination of MRD by flow cytometry. These two as if it is negative or very low value. And here the very low value is defined as lower than 1 in 10,000 or 0.01%. So they're exactly similar on the survival without leukemia. And at the same is the case for if any of these techniques detects that there is a residual leukemia in those patients. But if a patient has then the flow cytometry MRD determination as negative, but the DNA sequencing that is more sensitive detects some residue of the leukemia, that is the red line here, you can see that then you have a different survival free of leukemia. And we know if everything is positive for both techniques, it is bad. And we know if it's negative for both techniques, it is good. So it's better survival. But the red line shows you how significant it is. And at least for these 55 patients among 620 patients enrolled in these studies, this mattered. So this is why we can see that in the future, perhaps we are going to be able to standardize the HTS MRD and hopefully adopt in a way that we can be way more accurate on the determination of MRD. And furthermore, if you have then patients to whom the COG would consider as what we call standard risk of the leukemia coming back. So this means the leukemia, the characteristics of the leukemia for those patients at diagnosis were more amenable to the cure. If you have then the determination of MRD by HTS and it was negative, it was different from the ones with the positive MRD by HTS. And this was statistically significant. For patients with belonging to the so-called high risk chance of leukemia relapse and high chance of not surviving up to the leukemia, there was again a trend for a worse survival if there was a detection of the MRD by HTS and better survival for those with negative. But this, because it was so close, we couldn't see a statistical difference. So this remains to be studied better. But the idea that at least for the standard risk patients, we can see a distinction, this really emboldens the fact that we are getting to a very significant result. Talking about the risk of stratification for all the patients with acute leukemias, ALL, standard risk means if the patient was diagnosed between the ages of 1 and just below 10 years of age, and the counts for the white blood cells, they should be low at diagnosis. If the patient is above 10 years of age, or if they have a very high cell count on the peripheral blood, that means then the leukemia is more aggressive, and that means it's harder to treat. There are other factors that increase the risk of this leukemia to be more aggressive. One of them is the infiltration of the central nervous system. So for example, in the brain or in the optic nerve, or infiltration in the testicles, or at diagnosis, the patient has been already administered some steroids, for example, steroids for asthma or some sinus problem. That means this could affect the biology of the leukemia, because the steroids are part of the chemotherapy we want to give to these patients. Cytogenetics, these are studies based on the chromosomes of the cells, the leukemia cells. And this now we have learned a lot about it, and it is a better way to define what type of leukemia someone has. And they pretty much correlate very nicely with the behavior we see on the patients. Certain findings we get from the cytogenetics, you can even guess that would already belong to a patient either in standard risk or high risk. And now through the refinement of the MRD determination, now we even have learned how much leftover leukemia we want for a patient along the therapy. And this actually matters because the more leukemia cells we kill, the better, because that shows these leukemia cells are more sensitive to the treatment. So at the end of the first week of chemotherapy, we want to have less than 1% of the MRD positivity. At the end of the first month, or what we call the end of induction, we want less than 0.1. And at the end of a consolidation, less than 0.01. So now at the current trials that are open at COG for ALL patients, we have these refined definitions for high risk. So once again, we got these numbers from the previous studies, but even more. We have learned that even if a patient comes in with a high risk of the leukemia coming back, if they were classified as high risk just because of the high cell count in the periphery, but they are younger than 10, they didn't have any infiltration in the brain or the testicles. They have some findings on the cytogenetics that are characterized as favorable findings. And also, if they had good response, these are even though they belonged to the high risk group, we know they will have better chances of survival than the whole high risk group as overall. So this enables us to be able to define if we want to actually decrease the intensity of the treatment, to decrease the toxicity, and still get the cure of these patients. So now talking about immunotherapy. So as I presented, using chemotherapy, so all those drugs, all those poisons to kill the leukemia cells, that pretty much works in the favor of about 90% of the patients coming with ALL. But we have learned from all the other cancers over the last decade that if you can find a way to use the patient's own immune system to help us kill their own cancers, this is a different way to kill the cancer. And even better if the cancer is actually resistant to the chemotherapy. So that way you have a different way to treat that would hopefully rescue those patients to whom we didn't have other options. So belonging to this group of therapies, one of the drugs that came out is the blinatumomab, that it is what we call the bispecific antibody that works as an anchor or a fish hook to bring the T cells, that is the immune cells, to the blasts, to the leukemia cells. So that way the immune cells can kill the cancer. And another approach for immunotherapy is what you may have heard about, the chimeric antigen receptor T cells, so also called CAR T cells. And this is through gene transfer, so genetic modification of those cells. We render the patient's own T cells specific to the leukemia cells in a way that then the T cells will be able to kill their cancers. So nowadays, what we have is these immunotherapy approaches mostly for B cells. So if a patient has a T cell leukemia, this is still not available. So talking about then using the immunotherapy approaches, the COG clinical trial ALL 1331 was a clinical trial for patients with relapsed B cell ALL. So these patients had the disease coming back. So after they got in remission with the chemotherapy, the disease came back. So they randomized between either chemotherapy and then chemotherapy plus using the bispecific antibody blinatumomab. And the findings of the study they have already been published. And at least for the high-risk patients, the blinatumomab was superior for killing the leukemia and also to preventing the leukemia to come back. So because of that, now we have the new trial, the ALL 1731, now the proposal is really to give the blinatumomab also from the beginning. So instead of just giving chemotherapy, these patients are being randomized to receive either the chemotherapy alone or chemotherapy plus the blinatumomab. And because we know it has worked and it works on killing the leukemia, we are offering this all the way to even the standard risk patients. We do need to do this study because we don't know if adding something else to the chemotherapy may actually be more toxic. So we do need this very important trial so then we can learn better. And the hope is actually this way we will kill even more leukemia cells right from the beginning and hopefully then decrease the chances of this leukemia to come back even for those patients with standard risk. So still talking about immunotherapy approaches, then remember the CAR T cell therapies. The FDA has approved for using CAR T cell therapies only for those patients after the leukemia has come back after multiple rounds of chemotherapy. So this novel clinical trial 1721 is proposing to use the CAR T cell therapy for those patients with high risk to whom the MRD was still positive after about 2 to 3 months of chemotherapy. So that way we are already anticipating the rescue instead of waiting until the leukemia is really coming back in a vengeance. And another important trial that is also currently open, the 1732, also for high-risk patients, there is a randomization for these patients to receive the drug called inotuzumab ozogamicin. So this is a monoclonal antibody. So this is a man-made antibody that is against a marker that is on the surface of the B cells. And this antibody is conjugated to a drug that is a poison to these cells. So the antibody will direct the poison directly to those cells and hopefully this way decrease toxicity and activate the drug only in those cells that we want to kill. Another very exciting change we have made for the novel generation of clinical trials for ALL is the same length of therapy for boys and girls. So traditionally in the US, boys always received a whole year of additional therapy, so the girls would complete their chemotherapy earlier. That because in the older ages, what happened is these patients were at risk of the leukemia coming back earlier, perhaps because the leukemia was hiding on their testicles or in somewhere else in their bodies. But we have learned from multiple different clinical trials throughout the world that actually as we increased the intensity of the treatment using better drugs or higher doses of the same drugs, this wasn't happening anymore. So in Europe, for example, the length of therapy was already the same for boys and girls. So the US now has adopted the same trend with the idea that this is actually not necessarily anymore, but with the caveat this is still within the clinical trial. So we're still gonna learn from this. But the expectation is that now we really dialed down the toxicity and boys don't need to suffer longer than the girls. Now talking about T cell ALL, remember we were talking about the B cells. Now we are talking the T cells. So this is a different type of leukemia and we were always worried that these cells were harder to kill and to the point that they were more prone to infiltrate the brain and also to come back with a vengeance in the point that we couldn't get the same cure rates as we were getting with the B cell ALLs. So also the T cell lymphoblastic lymphoma that pretty much get the same chemotherapy course that the acute lymphoblastic leukemia patients. So we needed different approaches. And this different approach hopefully came embodied in this new drug, the nelarabine, that is actually not so new. The FDA granted accelerated approval in 2005 for patients to whom the disease with the T cells, either leukemia or lymphoma, had come back after at least 2 different prior regimens. And just published on the 1st of this month, the results of the AL trial 0434 that was completed actually in 2017. What we have learned is that using nelarabine together with the chemotherapy, the standard chemotherapy we use for all the ALL patients, we had decreased relapses without increased toxicity. Remember, I told you every time we add a new drug, we always worry that we are not going to harm more the patients than help them. And the best performing arm actually had a 5-year disease or leukemia-free survival of 91%. So this came up from about 75 to 80% all the way to 91%. And not only that, the patients who received the nelarabine had significantly fewer relapses in the brain. That's really what mattered to us, because that was the biology of this disease that we could see on the patients with T-cell ALL or T-cell non-Hodgkin's lymphoma. So now let's switch gears to AML, so myeloid leukemias. So this has been a real exciting few years for AML. So we always had the trouble. The AML is a different disease from ALL, and it's harder to treat. We need actually treatments that are way more toxic, that keep the patient in the hospital for most of the treatment, and with problems with requiring a lot of transfusions, a lot of infections, a lot of antibiotics. And after that, you would still could have this disease coming back. So we needed really new drugs. So the first one that came out, and actually wasn't that new as well, it was the gemtuzumab ozogamicin, or GO. This drug was actually previously approved for initial treatment for relapsed disease in the past, but because people felt it was toxic, too toxic, even the pharma company that made it withdrew the drug from the market, at least in the US. But in Europe, people still continued studying it, and actually they got it better. The gemtuzumab ozogamicin is, again, immunotherapy mediated treatment. It is an antibody against the CD33, that it is another molecule that is on the surface of the leukemia cells. And it delivers, again, a drug that actually is activated within those cells to specifically kill the cells that have the marker. And because of that, then, because it was so effective, people started new trials in the US. And we finally, in 2017, got firstly the FDA approval for the treatment of relapsed disease in children. And now, this year in June, we got the extension of the indication for even the patients with newly diagnosed AML, and all the way as young as 1 month of age. Another drug that is pretty exciting is the gilteritinib, and this is a different class of chemotherapy. It is an inhibitor to one gene mutation that is pretty common in AML patients. And this, when this is present, these leukemias, they are more aggressive and harder to treat. So using this drug, you can affect this mutation that really makes the leukemia more aggressive. And finally, we got in 2018 an approval from the FDA for treating adult patients with relapsed disease with the FLT3 mutation. So nowadays, the generation of trials that are coming down to children with AML is the incorporation of this drug for the treatment of those patients with the mutation. And another drug that has recently approved by the FDA is the CPX-351. This is what you can call as a nanotechnology drug. So liposome, so it's like tiny bubbles of fat that carry two of the drugs that we usually use for treating AML, daunorubicin and cytarabine, but they are carefully kept at a certain ratio in a way to optimize the effectiveness of these drugs. So the idea is giving these drugs within the liposomes, this is better than just giving these drugs in series to the patients with chemotherapy. Hopefully less toxic and more effective. And right now we have the approval of these drugs for adult patients with more aggressive AML, but the idea is we need to incorporate this into the clinical trials for kids. And this is exactly what the CRG has done. So this is a currently open trial, CRG AML 1831. So we are randomizing the standard therapy with chemotherapy plus GO, comparing to CPX 351 with GO. So you can see GO is the same thing for both because we know it works. So the main comparison is between the regular chemotherapy to the CPX 351. And on top of that, this clinical trial is also proposing to treat the patients to whom the FLT3 mutation is present, to also give the gilteritinib to really harness all the knowledge we have, the current knowledge we have for AML therapy in a way that we can really put everything to the test to really improve the treatment for these patients. And you can see here the same idea of the MRD determination. This is the end of the induction. So after the end of the first month of therapy, we already have proposed a certain number for as a marker of good response. If the number of leukemia cells or the residual leukemia cells, they are about this number. This is still to be tested, but the idea is we now know the fewer cells is better, but we need to define how much low is the good low. And another important thing that also harness the knowledge we have is the idea that for those patients to whom we define as low risk, and this again, it's very complex, but the idea is for those patients after the first month, if we know they have negative residual disease, and we know from the beginning from the characteristics of their leukemia that the chances of the leukemia coming back, they are lower, we can decrease the chemotherapies. It used to be years long, then it went down to about 6 months of therapy for about 6 cycles, and then 5 cycles. Now we're going down to 4 cycles of chemotherapy. There are 2 other classes of chemotherapy drugs. They're making a pretty much big buzz out there. Just yesterday, there was an announcement of FDA approval. One of them is the venetoclax that belongs to a group of drugs that inhibit pathways that promote the survival of the leukemia cells. And this drug is actually has been tested not only AML, but other types of leukemias and lymphomas. So just yesterday, the FDA announced the approval for venetoclax for adults, older adults with very high risk AML. And the venetoclax has an added advantage. Is actually a pill. It's not something you inject. It's not something that you need to stay in the hospital. Is just a medicine you take by mouth. So this is something that we are incorporating in the current and hopefully the future trials for AML, and hopefully it will be another drug. And another class of chemotherapy drugs that we're coming out as well, and it's currently being tested in adult patients, and we know it's very promising, is inhibitors of the IDH pathway. So these are growth hormones that work into the leukemia cells. So if you block that, the cells start dying. So we know it works in the laboratory. We know it works in mice. So now we're giving to people. And again, these are medicines you take by mouth. So all very exciting. Talking about Hodgkin lymphoma. So this is actually not very common disease, but when it comes down, it is pretty dramatic, not only because the person comes into the hospital very sick, but also because you can get rid of it very quickly. And in many cases, you don't even need to keep the patient in the hospital. But when the Hodgkin lymphoma is refractory to the treatment, or when it comes back, then it's way harder to treat. And this has been recognized as the ideal candidate for immunotherapy because within the tumor, there's a lot of the immune cells, normal immune cells that went into the tumor, but they weren't able to activate to kill the tumor. So we have learned that if you use some medications to reactivate these T cells, so what we call the immune checkpoint inhibitors, so then it works for these patients. So nivolumab and pembrolizumab, these have been approved for relapse disease. So we now know that if a patient has the Hodgkin lymphoma coming back, you can really use the immunotherapy for that. And there's again another drug, antibody drug conjugate. So this is an antibody that is specific to the CD30 marker. That is again a marker that comes into the Hodgkin lymphoma cancer cells. And it brings a poison that will then be delivered directly to those cancer cells. So this is a very good drug to treat, and it has been in many different clinical trials. So now the challenge is to see how you're going to combine immunotherapy and this new drug to treat these patients, either with relapse disease or maybe even with high-risk patients coming in. And another drug that is also in a lot of different trials, including children and young adults, is the bendamustine. This is a chemotherapy drug that kills a lot of these the lymphomas and the lymph cells. So FDA has already approved this for leukemias and lymphomas that show up mostly in adult patients. Now, again, the challenge is to really bring down to combine with immunotherapy to treat our patients. And for Hodgkin lymphoma, as I said, the cure rates are high. It's pretty much around 85, so 85 to 90%. So the challenge is you don't want to be more toxic. So we have these good drugs, so we probably keep in our pocket. We will give the chemo, the usual standard chemo, and only if you see the response is not coming very good, then you can make use of these drugs. So in summary, so we have very effective therapies. So now we are at the point that we are trying to decrease the toxicities to promote a healthy survival. We have seen a lot of expansion of indications of all these new drugs to children, in particular because there have been a lot of incentives to the pharma industry to include children in their clinical trials. And it is up to all of us to really make sure we have children being included in all these trials, children participating in all the trials, so that way we can know better and have more indications for curing kids. As we discussed, we are harnessing immunotherapy approaches. People say one day we are going to treat everything with immunotherapy and maybe even not give chemotherapy. Let's see. Right now, what we are trying to do, the main challenge is really try to integrate both the approaches. We have seen more focused research on childhood cancers, in particular because studying the cancers that come in kids, they actually provide more insight on the genetic determination of cancers. And some of the pathways, some of the metabolic pathways that activate those cancers, we don't see or we never noticed in the cancer cells coming from adults. So there has been a renaissance on the research involving childhood cancers, a lot of laboratories, a lot of financial support for doing research on childhood cancers. And similar to what we learned from the MRD determination, these studies are actually leading the way to better therapies and hopefully then translate to everyone and to every type of cancer to really improve the therapies and step confidently to a brighter future. So here I'm leaving my email, so that way if you guys need any questions, any clarifications, any further information, please feel free to contact. So now back to Lila.
Yes, hi. Thank you so much, Dr. De Oliveira. That was wonderful. And thank you for sharing your expertise with us tonight. Next up, we have the Executive Director for the Leukemia Lymphoma Society, Greater Los Angeles area. So Shannon Sullivan is going to be presenting next.
Yes, hi everybody. Can everyone see my screen? That is a yes.
Yes, but can you put
Yes.
up full screen?
Yes, does that work?
Yep.
Perfect. Okay, thank you. Well, hi everyone. Thank you all so much for giving me a few minutes to speak about the Children's Initiative. And thank you, Dr. Oliveira. It's so inspiring to hear. You are the person who, any technical questions, any research questions, I'm so glad he's on this call. I'm going to talk more from a strategic perspective about how the Leukemia Lymphoma Society is advancing cures and care for kids. So some of this will be a little bit of a repeat of what Dr. Oliveira said, but I think it's important to highlight it. We've made incredible progress on childhood leukemia. As you heard, almost 95% cure rate. And there has been quite a bit of innovation in new therapies, especially in immunotherapy and some targeted therapies. But leukemia remains the leading cause of pediatric cancer death with brain tumors. And you know, when we do have kids who relapse, you know, the statistics are not great on survivorship, and so it's important that we continue to you know, to look for new ways to treat this disease. You know, when we compare childhood leukemia to sort of the drug development that has happened in adults, you know, there have been dozens of drugs that have been developed to treat adult leukemia, and specifically acute myeloid leukemia and ALL over the last several years, but we haven't seen that pace of development for childhood leukemia. And as Dr. Oliveira discussed, the treatment regimen that most children go through is decades old, and it's highly toxic to kids. And that toxicity is really important to discuss because, you know, when a child is diagnosed with leukemia, it is an incredibly traumatic experience for the entire family. It's psychologically difficult. It's financially very difficult. So, and part of that goes back to just the sheer toxicity of the treatment, the chemotherapy treatment that is typically given. And that you know, while we have that 95% survivor rate, there is a cost. You know, when we look at the longitudinal studies of childhood survivors of leukemia, the treatment regimen that they go through does cause long-term damage to their bodies. So we see issues with organ damage, infertility, growth issues, and the list goes on. So, you know, clearly we can do better. So I want you to dream a little bit with me about what could potentially be possible. So what if we could develop drugs specifically for kids and repurpose drugs that are being used to treat other types of cancer and are already effective and FDA approved to treat other types of cancer? What if we can provide even more incentive to the drug companies to test drugs in kids and make it cheaper and easier and less bureaucratic for them to go through the drug development process? What if we could tailor therapies specifically to a child and rather than the one-size-fits-all approach, and reduce the toxic side effects by providing more targeted therapies for kids? So finally, what if we could get more cures and less toxic drugs to young people faster? Well, that's exactly what the Children's Initiative is all about. So this is the Leukemia and Lymphoma Society's very focused effort to drive cures and care for children. This is a $100 million initiative that was launched several years ago to really create a different way, kind of change the paradigm for drug development. That's sort of at the core of what we're trying to do here. There's really three major pillars of the program. So the first is provide more support and care for patients and caregivers. So we've increased our financial support programs. We've created new advocacy programs to, you know, help drive changes at the federal level. and at the local and state level. So very, very robust programs on the support side. We've also doubled our research investment. So Dr. Oliveira mentioned that there's more money going into research for kids, and LLS is a part of that. You know, we have to support the scientists and the researchers in academic medicine. We have to incentivize them to focus on childhood cancer. And so that's one of the important parts of what we're doing. But I think what's truly innovative about our approach with the Children's Initiative, it's our pediatric master clinical trial. And you may be saying, what is that? Well, you know what a clinical trial is. That's where we test drugs test drugs to see if they are effective in fighting a particular disease. A master trial is where you're actually testing multiple drugs. And so you have kids, you know, in this case, we are enrolling children in this clinical trial that have, you know, first diagnosed with leukemia. We're doing a full genetic profile of their disease. And then when they, you know, if they relapse, they have an opportunity to be part of the PEDAL clinical trial, which is the Pediatric Acute Leukemia Trial, PEDAL. So there's a couple things that are important to talk about with this clinical trial. So this was first envisioned in 2018, and we've been sort of putting the pieces of this in place since then. Children's Oncology Group is one of our big partners. But I wanted to talk about some of the things that we've accomplished since 2018. So first, we've put together an incredible list of partners. This is a global trial. We actually have 5 countries that are participating in this. We've gotten the governmental agencies, so the FDA equivalent in each of these countries, to agree that if a drug proves effective through this clinical trial, it will be approved in all 5 countries. That's important. Drug development, drug testing, clinical trials are incredibly expensive. And so this is a way that we're able to we're able to take that barrier away. So that incentivizes the pharmaceutical companies to be more engaged in our work and supporting our work and providing the potential therapies to be tested in kids. We have over 200 hospitals that are participating in this clinical trial that will get off the ground in 2021. We have, I believe, right now there's one trial arm that's been approved for acute myeloid leukemia, and there's several other that we expect to go to launch with as well. So pretty exciting, and we are thrilled to be leading this, but don't take my word for it. So actually, Lila has a great video that she's going to show that I think validates really the innovation behind the pediatric, the PEDAL initiative and the Children's Initiative. So Lila, if you don't mind showing that video, that'd be wonderful.
Can you see that? Can you see my screen?
What we're trying to do here with LLS PEDAL is to do something that's never been done before, and that's to match the child to a trial almost in real time. You could imagine a child coming in with relapsed leukemia and to be able to figure out within 24, 48 hours a new therapy to put that child on and to get that therapy started. That's never been done before.
We've been using the same drugs for the last 40 to 50 years. These are drugs that have been designed primarily for adult cancers, and we've been using them in kids to improve outcomes. And we've probably gotten as much mileage out of them as we can. For many of these types of leukemias that we're treating, we've pushed the limit of toxicity as far as we can. If we're going to make new progress, we need new therapies.
With this initiative, if we are successful, we can bring in massive number of pediatric leukemia patients to answer questions in a few short years rather than decades. And the fact that we have failed to do that really requires for us to change the way we're thinking and change the paradigm.
Pediatric leukemia is actually different from adult leukemia. So the changes that we see in the leukemia cells at the level of the genes are actually quite different from those seen in adult leukemias. So it's really important to actually focus research specifically on childhood leukemias, both acute myeloid leukemia as well as acute lymphoblastic leukemia. I am so excited that the Leukemia Lymphoma Society is going to support a next generation clinical trial very similar to the adult BEAT AML trial. This trial is actually going to enable a master trial for children with AML and other high-risk leukemias. So as part of this study, really for the very first time, we're going to be able to sequence the gene changes in a child's leukemia and then match those gene changes to drugs in a series of clinical studies.
Without LLS's backing, we could never even dream of a project like this, because this is a huge initiative that's going to require data from lots of different sources and an enormous infrastructure, the likes of which we've never seen before.
I have never been this optimistic as I stand here today. We can do this.
Wonderful. Well, thank you, Lila. I'm going to leave it to the experts. So you all are amazing. Dr. Oliveira, thank you so much. You're part of this, and I think that the most important thing to know is that we're going to find more therapies and more cures and less toxic therapies for kids, and that will happen over the next 5 to 10 years, and it can't happen fast enough for anyone who is treating children with cancer, leukemia specifically, or anyone who is caring for a child with cancer, or just cares about somebody who's battling this disease. I think we all know that we have to do better. So thank you, Lila, for giving me an opportunity to share the Children's Initiative.
You're welcome. Thank you so much, Shannon, for that. Really quick, we do have room for, I haven't had any questions come in, but we did have a question come in through the registration. So one thing, are there any clinical trials or research for pediatric AML with a 5q deletion? I'm guessing that's for Dr.
Yes. Can you hear me?
Yes.
Yeah. So specifically for the 5q, it's a little tricky because it's not very prevalent. So even we are still learning about the biology, but I can tell you, for example, the 1831 trial, a patient coming with a new diagnosis of AML that has the 5q deletion would be eligible for treatment in that. One thing that is important to remind you is a patient with that change in the cells, that could mean that it is more aggressive. So as what we know nowadays, this patient actually, even after getting to remission with the chemotherapy, most likely would need a bone marrow transplant, because the concern that the cells, the blood cells for that patient would be more prone to turn into leukemia, even after you control that leukemia that was at present at the diagnosis.
Gotcha. I have another question. At what point do you consider clinical trials when thinking about treatment for your child?
So as we have a very good chance of curing anybody coming through the doors of the hospital, there is virtually very few patients that we would at the first discussion just say, we don't have too much to do, you can go home, you can consider thinking about that. So the idea is there's at least about 1, 2, or for example, for leukemia, you probably have up to 3 different ways to treat the leukemia, even if the leukemia is resistant to the treatment, what we call refractory. So to the point that if a patient comes with ALL and the parents actually do not want to treat because they are afraid of the chemotherapy, for example, because we know we have high chances of curing this patient, we actually are mandated actually by law to treat the patient. So in that sense, the thought of having a patient going to a clinical trial would mostly be reserved to those patients to whom we notice, for example, the MRD, it's still there. We can't really clear the leukemia. Or if we treat the patient and you still see the leukemias, the leukemia cells in their blood, those are the patients we know very much likely that we won't find a chemotherapy drug to treat these patients. So that is why most of the clinical trials are actually reserved for when the patient comes in with a relapsed leukemia, even after 1, 2, or sometimes even 3 rounds of treatment.
Okay, and one last question. I know we're at time, but what is the latest with the use of nanoparticles with chemo meds?
So the idea for nanoparticles is that you can target the leukemia cells, so you can encase the medicine, for example, and deliver right there. Another way to understand nanotechnology or nanoparticles is in a way that you can increase the entrance of the drugs, not only by delivering or maybe opening holes in the cell or some other way. So in that sense, CPX-351 is a nanoparticle, is actually a man-made nanoparticle, but it is a nanoparticle. And most of these drugs in which we have the conjugation of an antibody and the medicine, it's also another way you could see as a nanotechnology, development of a nanoparticle, and depending on what you have. So there has been a lot of research using the laboratory models and mouse models, but the main challenge is really not only if it works, but also then when you go to make in large amounts to treat a person, then even if you have a good product, a good idea, if you cannot make a large number or large amounts of these nanoparticles, then you can't use. So that's why I would say it's a little bit challenging to really make the full translation into clinic. So there's still a little bit to go, but there's a lot of people studying that.
Good to know. Well, that is all the time that we have today. Thank you so much, Dr. De Oliveira and Shannon, for presenting today. So on behalf of UCLA Health and the Leukemia Lymphoma Society, I just want to thank everybody for coming. Take care, everyone.
Thank you.
Thank you, everyone. Have a wonderful evening.